continue our conference. And as you know, during the SARS-CoV-2 pandemic, topic of viruses is so important as never before. What do we know about viruses' origin and evolution? Let me introduce our next speaker, Dr. Koonin. Dr. Koonin leads a research group at the National Institute of Health in US. He has been working in the fields of computational biology and evolutionary genomics since 1984. His current research interests are coevolution of cellular life forms with genetic parasites, such as viruses, transposons, and plasmids, evolution and origin of viruses, and evolution of cancer. Dr. Koonin, we are very happy to have a chance to listen to your talk.
Thank you very much for the introduction. Good afternoon, dear colleagues. I hate the fact that I cannot see your faces. It would have been quite different. But I'm happy to be there nevertheless, imagining myself in beautiful Göttingen. And let's discuss the world of viruses. As our host so correctly noted, viruses have never been the realization of how viruses are truly important has never been as clear as in these difficult days. So what I want to present to you in this talk is an emerging global picture of the organization of the world of viruses and the new structure of the megataxonomy of viruses that follows from it. Reaching this understanding, or what I think is understanding, in the last few months has been rather surprising, to be honest, to my colleagues and myself, and hence this epigraph from Albert Einstein. The fact that the world is comprehensible is a miracle, in particular the world of viruses. To begin with, viruses are the dominant entities in the biosphere. The most numerous, simply put, physical entities and the reservoir of greatest genetic diversity. The number of viruses in all kinds of environments that surround us, as well as in those inside our bodies, is simply astonishing. 1 cubic centimeter of seawater, depending on the particular habitat, may contain between 1 million and 1 billion virus particles, which is typically 1 to 2 orders of magnitude greater than the number of cells in the same sample. So viruses are the most abundant biological entities in the biosphere. But not by mass, but by count, viruses dominate the biosphere. And the numbers themselves are totally astonishing. The body of each of us contains roughly 10 to the 16 viruses. And the entire Earth contains more than 10 to the 31st, difficult even to pronounce, 10 to the 31st degree viruses, which is greater than the number of atoms in the visible universe. So viruses are the most numerous and diverse biological entities on Earth. There are about 10 to the 24 stars in observable universe. Just as a comparison, to give you some kind of scale, and 10 to the 31st virus particles on Earth. Apart from that, viruses are remarkable in implementing in their reproduction different strategies of genome replication and expression. Of course, now everyone in the street knows that SARS-CoV-2 is an RNA virus. Perhaps not everyone is thinking about how remarkable that actually is, because in all of us, in each of our own cells, the traditional route of the transmission of genetic information takes place from double-stranded DNA, which is the genome, to RNAs, structural RNAs, and messenger RNAs, and from those messenger RNAs to proteins. This is arguably the biggest universal of all cellular life. Viruses are not like that. Viruses actually employ all thinkable, all logically possible modes of replication of nucleic acids, be it RNA or single-stranded or double-stranded RNA, or single or double-stranded DNA, or again, single-stranded RNA, but that replicates via step of reverse transcription through a DNA intermediate. So back in 1971, nearly 50 years ago, the discoverer of the reverse transcriptase, Nobel Prize winner David Baltimore, made the crucial conceptual step of organizing the information or processing cycles of viruses into 7 distinct classes based on the form of encapsulated virus genome. These 3 classes of RNA viruses: positive strand polarity RNA viruses, whereby the genome is the same polarity as messenger RNA for production of virus proteins; negative strand polarity, the opposite polarity, complementary; double-stranded RNA genomes, reverse transcribing viruses of 2 kinds. Some have RNA genomes and some have DNA genomes, but all use reverse transcription or synthesis of RNA and of DNA on an RNA template in their replication. And finally, DNA viruses, single-stranded and double-stranded. The double-stranded DNA viruses are overall apparently the most abundant in the biosphere, use the same strategy of replication and expression as cellular life forms. So we have to, I want to stress right now 3 more crucial facts or 3 more crucial issues regarding the nature of viruses. First of could it be that virus-like entities are nature's original genetic systems, in a sense testing different strategies of genome replication and expression? And finally, I want to emphasize, this is crucial for the rest, all of it, but this in particular, there is no common ancestry of all viruses, not a single universal gene among all viruses. This is in sharp contrast to the common ancestry of all cellular life forms that share about 100 universal genes. So in that respect, we're already starting to see that the evolutionary layout of the virus world is very different from that of cellular life. I also want to indicate that viruses of different Baltimore classes dramatically differ in host range. You just see that this Manhattan type landscape is very different in among viruses infecting different life forms. For instance, in archaea, there are only DNA viruses, predominantly double strand, to some extent single strand. On the other hand, in animals, actually, like us, actually RNA viruses dominate the landscape, and especially this is the case in plants, which is really a virus RNA world, and so on and so forth. There's interesting, still not fully understood interplay between these Baltimore classes, the different viruses, the different strategies of genome replication and expression, and the host ranges. I will now, in the next few minutes, I want to discuss a pressing issue, a very fundamental issue. Where do viruses come from? There are three principal concepts of virus origins. Researchers have been interested in the origin of viruses since the day they have been discovered, already about a century back, early in the 20th century. So, to begin with the beginning, virus early hypothesis, something that I have already alluded to. Viruses originate from the pool of primordial replicants that preceded the emergence of cells. The possibility is sort of hinted at, suggested by this diversity of genomic strategies. The regression hypothesis, something that I think historically appeared first. Viruses are the product of ultimate degradation of parasitic cells. Easy to explain. Finally, escape gene hypothesis. The scenario in which viruses are descendants of combinations of cellular genes that somehow have gone berserk and became autonomous parasitic entities. So these hypotheses have been mulled over in the literature for decades. Where do we stand now? Well, now with the advances of genomics, we have genomic sequences for a huge diversity of viruses. Now we will come to that. We now have the tools to actually investigate the origins of viruses, not in the realm of speculation, but very specifically using concrete data. And the key type of data for this are what we call virus hallmark genes, genes that include key proteins involved in virus replication and morphogenesis and are present in diverse viruses that span more than one Baltimore class. Each of these is very interesting proteins. I could talk about it for half an hour easily, about each of them, and the evolution and diversity. But we cannot do that. We have to be very quick. So these are, for example, so-called single jelly roll capsid protein. Typical capsid protein of RNA viruses and small DNA viruses, we will encounter it. Double jelly roll. Double jelly roll capsid protein, sort of a doubling of this domain found in a variety of DNA, great variety of DNA viruses. The so-called super family free helicase, the type of helicase that for some reason is found in a great variety of diverse viruses. Very importantly, RNA-dependent RNA polymerase and reverse transcriptase, quintessential of viral enzymes that you do not really find among normal cellular genes. Rolling circle replication, initiation, and the nuclease, another very, very distant relatives of these proteins. There are few of these hallmark genes. I show here the most prominent ones. There are more, about as many more, but they're our key windows into the evolution of the virus world. Some of my colleagues have investigated that in great detail and came to some fairly simple but very striking conclusions, namely that the replication modules of viruses have very deep evolutionary roots. The same structural fold, the so-called RNA recognition motif, a very simple, very robust fold, underlies the replication modules of all viruses. To the very best of our understanding, this is one of the most ancient folds in protein folds in nature that is very likely to have been one of the first protein folds emerging in the RNA world first as a facilitator of RNA replication by ribozymes, then evolving, diversifying into different forms of enzymes that are involved in the replication of virus genomes. The roots of virus morphogenetic modules, or the structural proteins of virus particles, however, are not so deep. With my colleague Mark Krupovich, we managed to show that we can trace ancestry from distinct cellular proteins for the great majority of the major structural proteins of viruses. Hence, this unifying, so to speak, chimeric scenario of virus origins. Chimeric because this is a blend of two of those three major scenarios for the evolution of viruses, namely the virus's early scenario and the escape gene scenario. The replication modules of viruses evolved under the first of these scenarios, under the primordial scenario, come from the primordial pool of replicating genetic elements, whereas the structural proteins can be grafted onto those replicons under the escaped gene scenario. The combination of these two routes of evolution with the capture of structural proteins occurring at very different stages of evolution resulted in the known in the world of viruses that we know. All right. So much for the origins. Now I switch gears and we'll talk somewhat more methodologically about metaviromics, search of viruses in metagenomic data, which is truly a tectonic shift in our understanding, led to a tectonic shift in our understanding of virus evolution. Just in case, metagenomic is sequencing of all DNA or RNA molecules from a particular environment. This has become an enormously powerful methodology in the last few years. And moreover, not just enormously powerful methodology, this is now, metagenomics, is the main source of the discovery of novel viruses, as we shall see very shortly. This is so prominent that 3 years ago we got together with a group of colleagues and came up with this consensus statement according to which now new viruses and new taxa of viruses can be officially recognized by the International Committee for the Taxonomy of Viruses without any biological characterization. In the field of taxonomy, this is a revolutionary step, but it was inevitable because an increasing great proportion of new viruses are discovered by metagenomic analysis. You can see how it looks at this slide that is about 3 years old, and the picture has become even far more striking since then. But even here, you can see that these colored circles that represent viruses with known hosts isolated from the ocean are just a minority of viruses discovered. Moreover, the most prevalent, found in the greatest number of places on Earth, and the most abundant, that is found in greatest amount viruses, are all metagenomics. So metagenomics now rules. It certainly does not cancel and never will the importance of biological characterization of viruses, but it rules the discovery of new ones and hence our exploration of the virus world. So let us talk about our exploration of the virus world. We don't have that much time then, but I'll try. First, the world of RNA viruses. Dramatic expansion in the last couple of years. Again, metaviromics, which reveals enormous diversity of RNA viruses in the environment and greatly facilitates reconstruction of the global picture of evolution of the RNA viral. Early in this talk, I emphasized that viruses do not share a single or universally conserved protein. And this is absolutely true if we talk about all viruses together. But major groups of viruses do share conserved proteins, fortunately for us. Among the RNA viruses, there is only one such protein, the equivalent of 16 sRNA in cellular organisms, if you will, and this is the RNA-dependent RNA polymerase, RDRP for short. Importantly, the RDRP is also a homologue of reverse transcriptase in reverse transcribing viruses, so we can build a joint phylogenetic tree for these virus hallmark genes. I will skip I must skip, basically, to my regret, perhaps, the methodological details, which are somewhat difficult because these are highly diverse enzymes. So we had to develop an iterative computational pipeline to come up with the big tree. We did that and came up with it at some fairly coarse level of resolution. We got a tree of about 5,000 RNA-dependent RNA polymerases and reverse transcriptases. At that point, a couple of years ago, when this particular analysis was done, about half of these marker proteins come from metaviromics. And we resolved the structure of this tree into 5 major branches that are quite stable with regards to various kinds of tests, the choice of sequences to analyze, the method of tree construction, etc., etc. Two of these branches include solely positive sense RNA viruses. One includes positive sense RNA viruses and a large group of double-stranded RNA viruses within that branch. Another consists entirely of double-stranded RNA viruses, and finally, from the inside of that branch, like birds from the tree of dinosaurs, springs the branch of negative sense RNA viruses. And I would like to emphasize that this tree encompasses viruses infecting bacteria, like here, this first branch, this deepest branch, as well as all kinds of eukaryotes. So far, no archaea. So of course, in this tree, I could not refrain from mapping the common parasites, including the coronaviruses that now are the reason behind the fact that I have to speak to you remotely, as well as flu and Ebola. You see that the position in the tree is very well defined. They belong in different parts of it, though. So there are several major conclusions coming from this analysis. These 5 stable major branches that, as you will see, become the highest level taxa of RNA viruses, and distinct relationship between positive sense RNA viruses and double-stranded RNA viruses. Basically, this is sort of like a Russian doll. Double-stranded RNA viruses evolve from the bin positive sense RNA viruses on at least 2 occasions, and negative strand RNA viruses apparently above from the bin the double-stranded ones. We also determined that evolution of positive sense RNA viruses, the largest part of the RNA virus world, occurs to a very large extent by accretion of domains, of distinct domains, such as helicases, proteases, and which are most common, and quite a few others, leading to increasing complexity of the virus genomes. And very notably, the infamous coronaviruses have the most complex RNA genomes with a great variety of different interesting domains accumulated in the genomes that reach up to 40 kilobases, the largest, more or less by far, genomes among RNA viruses. In a very recent metagenomic project, we obtained what I find to be an astonishing result. I still cannot go over it, or cannot get over it, because in the virus fraction from just a single metagenomic sample, or a large one, but from a rich habitat, but just a single metagenomic sample collected here in the estuary of the Yangtze River, we obtained a doubling of the known diversity of RNA-dependent RNA polymerases, which suggests, of course, that the entire diversity of RNA viruses on Earth is just humongous, enormous, and we are not seeing any kind of saturation just yet. At the level that roughly corresponds to a genus of viruses, at the level of relatively precise sequence resolution. At the higher level, however, and this is the other side of the coin, all these viruses, we have not discovered a sixth branch of this tree. All these novel viruses fit into one of the 5 branches. Within those 5 branches, there are very interesting major groups, novel groups discovered that become new high-level taxa. We also review the ancestral relationships between viruses that appear to infect unicellular eukaryotes in these habitats, and well-characterized animal and plant viruses. They finally, in a sense, know the ancestors of these viruses in unicellular life. And we are in a position to make the general conclusions on the RNA viruses. So these 5 new file of RNA viruses corresponding to the 5 branches in the overall phylogenetic tree of RNA-dependent RNA polymerase. Positive sense RNA viruses, the simplest genome replication expression strategy, are, it's clear, the most ancient RNA viruses that begot double-stranded RNA viruses, twice at least. The one lineages of the double-stranded RNA viruses giving rise to negative sense RNA viruses. The evolution of viruses, of these viruses, included acquisition of a variety of domains at the very, very early stage. We believe acquisition of the capsid protein that then had been replaced on a number of occasions by different types of gene proteins, acquisition of helicases that allows the expansion of genome, and acquisition of many other domains, and displacement of these domains by the functionally equivalent ones, which results in the observed diversity. The last few minutes, an impossible task, a whirlwind tour of the rest of the virus world. Primarily, I cannot discuss the details. So primarily, what I want to convey here is that we have clear-cut evolutionary scenarios for all other parts of the virus world as well now. In particular, for the reverse transcriptase virusphere, which includes such very well-known viruses as HIV, as well as endogenous retroviruses. The integrated remains of this comprise a very large part of our genome. Single-stranded DNA viruses, very interesting, very remarkable, small DNA viruses that we are now convinced evolved from single-stranded DNA plasmids by capturing, on several independent occasions, capsid proteins from RNA viruses. And finally, the double-stranded DNA virus pair. The most complicated one, where we do not have a single universal evolutionary marker, and therefore have to turn to different methodologies of analysis, in particular analysis of networks of gene sharing, to reveal the internal structure of this part of the virus pair. And we did that successfully, I would say, and have now a clear idea of the evolution of the DNA virus pair from relatively simple virus and relatively small viruses infecting bacteria in their archaea, to the huge, including giant, famous Mimi viruses, Pandora viruses, as well as such key pathogens as pox viruses, smallpox, that infect all kinds of eukaryotes. I'll briefly mention that these famous giant viruses, which generate all kinds of speculation about four domains of life and reductive evolution and whatnot, actually we can show very clearly how they evolved from smaller viruses by gradual accumulation of genes. Let's leave that alone and conclude the global architecture and megataxonomy of the virus world that earlier this year has been approved, to my astonishment, I should say, by International Committee on Taxonomy of Viruses. So we have these four highest taxa: the riboviria, RNA viruses, and reverse transcribing viruses, monodnaviria. We have to follow Linnaeus and use these Latinized names. No way out. Single-stranded DNA viruses and two distinct realms of double-stranded DNA viruses. One called diplodnaviria, which includes tailed bacteriophages and herpes viruses. One called varidnaviria, because there are various double-stranded DNA viruses that include the rest of the giant viruses and beyond. For each of these realms, we have developed the detailed taxonomic structure that is currently formally accepted. Of course, here, for the double-stranded DNA viruses, there is an enormous amount of fascinating discoveries to be made. And therefore, the internal structure will evolve. For instance, I just mentioned one. We have now investigated in detail the so-called crust-like viruses, bacteriophages, the most abundant, most common viruses inhabiting our bodies, our guts, and discovered solely through metagenomics, now finally confirmed by biological analysis. So a lot to be done here, diversity to be expanded dramatically, but I think the realm and the kingdom stand. Ditto for the other realm of DNA viruses, varidnaviria, extreme diversity already known and classified, a lot remaining to be discovered. So we can combine all these into a global network of evolutionary relationship in the virus world, these four monophyletic divisions, realms, involved in a somewhat complicated relationship with the Baltimore classes, with the classification by genomic expression and replication strategy, whereby some of the realms, such as the riboviric, include more than one Baltimore class, both the three classes of RNA viruses and reverse transcribing viruses as well. And on the other hand, some other Baltimore classes, such as double-stranded DRNs, split between at least two distinct realms. That's about it. I will just want to mention very quickly that many discoveries, great many discoveries, probably more than we have already made, are being made in different parts of the virus world. At the same time, I think we understand it. It's a bold statement, but I'll make it. I think we understand the general layout of this world. We may be settling on a stable evolutionary scenario of virus evolution and the megataxonomy of the major branches. At smaller scale, though, diversity seems to be huge and very habitat specific. There is a long path to saturation. There's a lot of fascinating discoveries along the way. So there are many tasks here remaining for biologists. I don't think they are going to be out of work for decades and decades. One of which I mentioned last, are deep reconstructions, reconstructing the viromes of, say, Lika, the last eukaryotic common ancestor, and Luca, the last universal cellular ancestor. And that last task we have undertaken recently, coming to interesting conclusions on the ancient complex virome in this paper that unfortunately I don't have a chance to discuss now, but perhaps some people will be interested. So my acknowledgments to my colleagues, without which nothing would have been possible. Valerian Dole, my collaborator of many, many years, decades, primarily in RNA viruses, but also beyond. Mark Kropovich, from Institut Pasteur, my major current co-conspirator in the study of evolution of the virus world, Yuri Wolf and Natalia Yutin, senior scientists from my laboratory, have been key to these discoveries. Several other colleagues who have been involved in the taxonomy development. And thank you all so much for listening. It's a pleasure talking to you. Thank you very much. Hello?
That's the questions. Yes. Okay, we got some questions.
Hello?
Yeah. Hello, can you hear me? Hello? Dr. Koonin?
Yes, hello.
Yeah, okay, yeah, good. Now there was some connection problem, I think. Okay, the first question.
I hope not during the talk.
No, not during the talk.
All right.
Just now. So the first question is, logic of chance was almost 10 years ago. What is your current stand on the region of eukaryotes in light of the new data? The question is from Maya Polovitska.
Of course, I very much appreciate the question. The reality of it is that I cannot really cover it, but I will say a few words. It's very true that the logic of chance was a decade ago. I'm trying to slowly get into the next version of it, which will be much transformed, far more elaborate, hopefully. So, regarding the origin of eukaryotes, there has been a major, this will be very brief. We're actually working on a major paper addressing the issue now. What happened since then, the main thing that happened, has been the discovery of the Asgard superphylum of Archaea, which carries an astonishing variety of homologs and likely ancestors signature eukaryotic genes, particularly those that are involved in membrane remodeling and cytoskeleton. And apparently they're phylogenetically related to eukaryotes. So I think the winning current wisdom is that eukaryotes have evolved from within the Asgard superphylum of Archaea from a particular branch, and then, of course, early on captured the bacteria and the symbiont that became the mitochondria. However, I would like to note that, in my opinion, the jury is still out on the actual scenario of the origin of eukaryotes and on the archaeal ancestry. The Asgards are very interesting. They definitely have a relationship with eukaryotes in terms of those signature genes. But I think the more realistic scenario could be more complex than what I just described. And this is what I'm going to say because we don't have all the time in the world. And this is not quite ready even, but stay tuned. I think there will be lots of new developments and fairly soon.
Okay, then we'll proceed with the next question. It's from Hanna Rochke.
Hello.
Hello, Dr. Koonin, can you hear me now? Hello? Yes. Oh, I think we might have again some connection problems. Hello? Yeah, okay. Dr. Koonin, are you back?
Yes, hello.
Okay, good. Yes, so we have a question from Hanna Rochke. She's asking, in metagenomics, how one can distinguish between actual viral genomes and viral insertions.
Yeah, okay, please repeat. There was a momentary interruption.
Yeah, in metagenomics, how one can distinguish between actual viral genomes and viral insertions.
Between the genomes of free viruses and those integrated into host genomes. Interesting question. There is no single universal answer. It has to be noted, first of all, that a great number of viruses do not integrate into host genomes. For instance, for all RNA viruses, this is not, except for the reverse transcribing ones, this is not really a problem. If they integrate, this very accidental and not really important. So on many occasions, we simply do not have the problem. On other occasions, such as bacteriophages, the problem is very much there. It's sort of alleviated when people analyze the virus fractional virus particles. It's easier. Also, however, in much of our work and that and others recently, we look into genomes that are virtually circular. They don't have to be physically circular, but the assemblies look circular because of terminal redundancy, which is the prevailing genome structure in double-stranded DNA bacteriophages, where distinguishing between free viruses and prophages is particularly important. So we have a tool to do this, these circular assemblies. On other occasions, though, admittedly, there can be uncertainty. You also can look at neighboring genes, and if there are no clear cellular neighboring genes, then this is probably a free virus. Hello?
Hello? Yeah, okay, we can proceed now to the next question.
Please.
There is a question from Yaji. She's asking about whether there is some evidence supporting the virus origin hypothesis. I think she wants to know about some experiments or whether they can be performed.
Well, there is, I would say, a huge body of evidence from comparative genomics. Performing experiments on the origin is highly challenging. I don't think that anyone is really addressing this. In principle, one can.
I think we got again some problems with internet connection. Okay. I think we need to wait a little bit.
From cellular hosts. But this is not something that you
Okay.
obtain easily. I think some very clever experimental setups, considerable amount of time are required to even have a chance. So this is. I don't think that this is an experimental plan now. It's a dream.
Oh, we just got again some internet problems. I think so. Could you please repeat? I think the last of couple of your sentences.
Yes, I can. So what I said was that these scenarios are based on a large body of comparative genomic evidence. Obtaining experimental evidence is very hard when it comes to the origins of anything, for that matter, origin of life, but origin of viruses too. So I can dream of experiments where the capture of capsid proteins by, so to speak, naked replicons occurs in the lab, but these will be coming up with a realistic experimental setup is highly challenging in this case.
Debujit is asking why do RNA viruses show extremely high rates of mutations over DNA viruses?
Yes. So the simplest answer is that RNA viruses, the great majority, the only known exception, by the way, of coronaviruses, have no proofreading nucleases. They don't proofread the replication process, hence very high error rates. Of course, the inquisitive minds will immediately ask, and why not editing, not proofreading activity? There are different answers, potential answers. We don't know why, but there are thoughts in that regard. One is very, very simple. The RNA viruses have small genomes, apparently because of the physical properties and chemical fragility of RNA, and therefore they do not very easily accumulate new genomes. They only accumulate new proteins and domains. They only keep those that they really need. Apparently, proofreading nuclease is not one of them. But there can be a more interesting answer. And that is that the so-called quasi-species structure, the diversity of RNA virus population, is actually important for the evolution. There is some insufficient, but some distinct experimental evidence to that effect. So perhaps even supposing that at some point of evolution, an RNA virus captures a proofreading nuclease, it's not kept there by selection because it brings counterintuitively disadvantage rather than advantage.
We can then proceed to the next question. Mariana is asking how reliable are reconstructions of virus phylogeny based on gene networks, and what are the challenges there?
How reliable are virus phylogenies based on gene networks, and what are the challenges there? First of all, I wouldn't, from the networks of the kind that I have been showing from gene sharing networks, you do not have you do not derive phylogenies per se. You derive classifications, clustering that seems to reflect evolutionary relationships. And with that qualification, I dare say that the major clusters reviewed in that manner are quite reliable. They show a lot of evolutionary coherence judged by other criteria, biological criteria, variant structure, and more. Within each of these, you can build also phylogenies that bring quite coherent results. I think these are these analyses, given that with another important qualification. That has to be done for large viruses with many genes. In such a case, they are reliable. They are not particularly useful, these methods, for small viruses or any other small genetic agents with a small number of genes. Challenges, I would say, comprehensively include one challenge, as I said, is some genome size. The other, more pertinent actually, even for large viruses. Now, there are many viruses that have diverged very far. So comprehensive and accurate identification of homologous genes is an important underlying challenge.
Then I think I will also take an opportunity to ask a small question. My question is how much the proteins has got changed? Do you think after they got incorporated into viruses, according to your theory? Is there a chance to find some completely new enzymes, for example, in the virome?
Or is there a change to find some completely new enzymes, or let us say more general, completely new protein folds? The answer to this is yes, and we do find such new protein folds, sometimes even enzymatic, but it has to be understood in context. And the context is that these new things that may even emerge de novo in viruses are not particularly common. I think we know the common ones. We know the hallmark genes, we know their structure, we know what they do, they know pretty much how they work. But among proteins that are present in narrow groups of viruses, there is enormous diversity, including things that are indeed completely new.
Okay, we are running out of the time. Thank you very much for us answering all the questions, and thank you very much again for your
It's
talk
a
and
pleasure
being together with us.
to receive so many questions. It's really a pleasure to receive so many questions. Stop answering them because we are running out of the time rather than for other reasons.
Yeah, if you would like, you can also join us tomorrow for our informal meeting at 8 p.m. Right? At 8 p.m. If you will have time, of course.
I'll look into this if I have a chance. I very well might join, and there might be perhaps additional questions.
Okay, then thank you very much. And
All right, thank you so much. My pleasure.